Great. All right, so thank you everyone for coming. Today I'm presenting a little bit of work that's been done for one of my dissertation aims, and this is actually sort of an information session for everyone here because I'm hoping most of you will be able to help me for some data collection to complete this aim, finalize it, and get some interesting information. I'm very interested to see what everybody thinks. So specifically, I'm going to be talking about approaches to visualization of harms data in clinical trials. And there we go. Just a little bit of background. When I talk about harms, what am I talking about? I'm really using it as an umbrella term for negative effects of interventions. So this encapsulates adverse events, adverse effects, side effects, complications. This is an appropriate term, general term for the concept in both clinical trials and evidence synthesis. And this is supported sort of by Consort and Prisma harms extensions. Harms are classified according to standardized and hierarchical language. So COSTART, MEDRA, CT, CAE, and they're also classified according to other metrics like rarity, so common, uncommon, rare, very rare harms, and severity, so harms that are mild, moderate, or severe, or serious, which has its own sort of regulatory distinctions. To clarify, when I say a hierarchical system, something this is important for harms and you'll see comes into play later on, I want to really explain what I mean by this hierarchy in case anybody isn't familiar. So here's an example of MEDRA classification for nasopharyngitis. The preferred term is the lowest standardized term for a specific or unique harm, and below that are the lowest level terms that map to the preferred term. So these are classified as the preferred term. So these lowest level terms, if you think about it, are really sort of the entry terms, or they're the common ways that people talk about harms or what they might have, what might be entered into a patient's chart. So maybe somebody says they have a cold. Analyses at that lowest level is really not advised because terms are really describing the same thing. You have different terms for the same thing. So if you try and do analysis there, you're going to be undercounting your events, what the actual event is, leading to an inappropriate estimate and statistically a big loss in power. So really the lowest level of harms for analysis you should be looking at is preferred terms. Preferred terms are organized then according to higher level and higher order terms. These are things like mid-level body systems or organ classes, and these are groupings of the preferred terms that share that whatever characteristic it is at that level. So for example, this nasal pharyngitis, nasal vestibulitis, these things all fall under viral upper respiratory tract infections, which falls under respiratory tract infections, falling under the system organ class, respiratory, thoracic, and mediastinal disorders. So when preferred terms are aggregated and analyzed at this sort of higher level, it can give you a sense of the general types of harms or body systems that might be most affected by an intervention. And there's also a further increase in power associated with this. You're essentially creating a large composite of specific harms that have a shared characteristic, but you also lose out on the details of describing the specific harms that might be of interest if you do choose that higher level only for your analysis and presentation. So why are we interested in visualization? Now that we understand a little bit about harms, why do visualization? Why not just have a big table? So one reason is harms data are incomplete and truncated for publication. So this is a big problem. They're often presented just as counts, and then you get a trial publication that has a table of, say, the most common adverse events that were recorded, the most common harms, and they're pretty highly selected because they meet specific selection criteria, which I'll get into. The other reason is that consumers, patients, clinicians, people who want this data, they want more information than they're getting in just that table of counts. So to really understand the full picture of potential harms, people need other aspects of the harms than just how many happened. So for selection criteria, when we see a report of harms in publications, it's never the full set that were collected. There's just too many to include in a table in the publication itself. So authors apply selection criteria, which are these thresholds and rules that dictate which harms of those collected get reported. So this figure shows the proportion of collected harms that would be reported if articles used a given criteria. So outlined in the black box is a criteria that was used in Rice 2001. This was a trial of gabapentin for neuropathic pain. And using that criteria of 5% of all participants combined across groups with no difference in frequency threshold, if you compare those harms reported that way with the unpublished data, the full unpublished data of all the harms that were collected across the trial, only 4% of collected harms were reported. And while some types of figures can become muddled by including large numbers of unique harms, there is the potential to circumvent a very strict selection criteria, present more harms, present a more complete picture of harms collected in a trial in a figure than in a table. And part of that reason why figures are better or can be better than tables is because sort of this other issue of multidimensionality of harms that have wanting more information than just the counts. So there are other aspects of harms that might be of interest to consumers. Most harms are generally mild or moderate in severity, and many preferred terms can occur at sort of several different levels of severity, so having allergic reactions that are mild versus life-threatening or deadly. But then for a given intervention, you may have some harms that have differences in timing or recurrence or duration. These are also sort of things that you need to consider if you want to take an intervention, or you may want to consider. So some harms, for example, may appear more quickly than others after very little exposure, whereas others take a long time to develop. So that can be a factor of dose and duration of exposure. Some harms may appear only once and then resolve completely, whereas others may occur multiple times. And additionally, some harms might be similar in those dimensions, but have different patterns of duration. So, maybe you have headaches that begin shortly after exposure to a drug and occur as multiple brief intermittent instances over several days, or you may have waves of headaches that last multiple days and begin shortly after exposure. So there's these different aspects of harms data beyond just the count, how often they appear. Now, visualization approaches, what do we mean, and sort of what are all the different ways that visualizations may be used to present harms data? So this is a recent scoping review that was completed by Rachel Phillips and Victoria Cornelius, both of whom I believe we have on the call, and both are working in a very similar field, sort of this exploration of harms and trials. So we're very happy to have them as collaborators on this project. In their review, they were specifically looking to assess all of the different analysis methods really for harms in clinical trials. And part of that was these visualization approaches, ways of summarizing visually harms that are collected. And on this figure, you can sort of see there's 20 methods for visual summaries. So of these 20 methods that are noted in the figure, there was another breakdown by application, and really only a handful were intended for the purpose of presenting data of multiple emerging harms, which is what we're really interested in exploring in our trial setting, what we're sort of envisioning for presenting an overall summary of all the harms. And then of that handful as well, there were a couple that were described in the original source material, but didn't have visual examples that could be reproduced to get the original author's intent. So that left us with 5 types of visualizations that had examples that we could replicate with our own data and that we were interested in examining more closely. So we took the 5 visualizations we could reproduce, dot plot, bar chart, volcano plot, heat map, and tendril plot. And we've recreated them using individual participant data from 6 trials of gabapentin for neuropathic pain from the MUD study. These are trials that we have the full clinical study reports for and the harms data for. In the process of writing the code for reproducing the visualizations, I actually identified sort of a 6th visualization it's a subset of the heat map, the Treemap. And so even though it may not have been included in the scoping review because it really is a type of heat map and scoping review looks at concepts as opposed to every single specific possible iteration, this particular visualization, I've been told, has been used for harms internally by the FDA. So we wanted to include it as well. It's different enough from the original heat map, and it's something that we know also sees use. So we wanted to compare it as well. So far, we've already sort of examined the characteristics of these visualizations. We've written up some results about how they compare related to their generation, like, which ones need IPD, individual participant data, which ones are created from aggregate data, the code that's needed to create them. But the last part of data collection that we need involves a comparative assessment of value for these approaches. And we've submitted a letter of determination for the Hopkins IRB, and this is non-human subject research, and it's where I'm hoping to get help from all of you. So we're going to get to that shortly. But right now, I'm just going to introduce you to each of the visualizations and spend sort of a minute just to describe what it is each of these visualizations is presenting, so that you are familiar with them as if they were being presented in a journal article. So the first one up is the dot plot. And the dot plot is a presentation of harms data at the level of the preferred term, arranged by descending effect size, and includes both an estimate of incidence in each arm and a measure of effective choice. So in this case, relative risk. Every recorded harm we've got shown here, and the summary estimates include the 95% confidence intervals. The measure effect is presented on logarithmic scale on that x-axis, and the green line indicates boundary of no effect. The bar chart, a little bit in contrast, it presents data on harms only at a mid-level body system, so an aggregated higher level than the preferred term. And it displays the number of occurrences within each body system by treatment arm. So here it's really just the count. But within that, it does break down the severity of the harms. So you can see the distribution of severity within each body system, and also as presented by those different colors. Volcano plot. This one presents all the harms again at the preferred term level, and these are, each term is represented by a bubble. Y-axis is the negative log 10 p-value, which we chose to use unadjusted, but you could use adjusted. So the higher you go on the y-axis, the smaller the p-value than ones lower on the y-axis. And on the x-axis is measure of effect, risk difference in this case, for gabapentin versus placebo. So you can sort of see there's this V, and that makes sense. At that V, you've got 0 at that point, which is no effect. And to the right of that is harms that are more common among gabapentin. To the left is harms that are more common in placebo. That's also codified by color. And then the size of the bubble is proportional to the total number of events. So the total number of events in both arms that were captured. The heat map. So this heat map presents data for subsets of harms. So overall, and then harms that meet specific criteria of interest. So say only harms occurring in males or females, and only severe or serious harms. And that data, the effect estimate, which in this case is a standardized risk difference, is presented at the preferred term level. but it's also giving you some data on midterm at the higher order terms, because the preferred terms are grouped according to their mid-level classification. The preferred terms are ordered in their intensity and their standardized effect, and the mid-level systems are also ordered by absolute count. So you have at the top, you have the body system with the most number of occurrence in gabapentin. At the bottom, you have least number of occurrence in gabapentin. And then here's that alternate, Heatmap alternate, the Treemap. So this presents overall harms at the preferred term level, again, for standardized effect, risk difference, arranged by their mid-level terms. So here again, it's sort of giving you some information on mid-level as well. And the size of the boxes for each harm is proportional to the number of events in gabapentin. So the size of each box and then the size of each grouping really tells you which harms are occurring more often in gabapentin. The last plot is the Tendril plot, and this is also sort of the most recent one. 2018 was the paper that came up with this. This presents all harms at the preferred term level. So each tendril is a different harm. And the tendrils are created of vectors. So the point at which they all start, so the tendrils all spread outwards from a point of basically what represents the randomization, the point of assignment to the intervention. So X and Y axis don't really have as much a purpose. Instead, the vectors dictate how the harm, sort of the pathway of how the harm occurs in the trial. So the length of a vector is the time between each event. So very small vectors, time between each event is very short, longer is longer duration. And the direction, whether it goes to the left or to the right, clockwise or counterclockwise, is the arm in which it happens. So an event in the intervention arm, gabapentin here, will angle it counterclockwise. And each point represents an event. The color is the adjusted p-value for that specific event, and the size of the points, again, is the overall count, the absolute sort of count for the event. So you can see all of these visualizations are sort of presenting different aspects of the data on harms, but how can we assess the relative value of these approaches? So what is value? Value is a measure that goes beyond the ability to support answering questions about the data. It centers upon a visualization's ability to convey a true understanding of the data, a more holistic, broad, and deep innate sense of the context and the importance of the data in the big picture. So the way that one would, sort of the gold standard for assessing, maybe which visualizations are valuable, is quite intensive. That would be, taking domain experts to develop baseline goals or tasks, things for the visualization to convey to a group of people, gathering a representative sample of users to accomplish those desired tasks, see which visualizations are able to convey that information, assess how the users then feel about the visualizations and maybe determine what needs to change to meet the user's needs. But that kind of assessment is logistically challenging, very time and resource intensive, and sort of beyond the scope of what we had for this project. So something that was given to us, suggested as an alternative, maybe not complete alternative, but for what we're doing, and then to later inform a further evaluation, is this approach that I'm going to ask all of you to help me with. This is the ICE-T, and this is another quite recent development, sort of 2018. This paper describes this new approach to assess the value of visualizations that's quicker and less resource intensive. So it's really based upon this premise that the value of a visualization is made up of several different components, sort of the time that it saves you, the insights that it gives you about the data, and questions you can answer about the data, the overall essence of the data that it conveys, and how confident in the data you are about the information that it's giving you. So the ICE-T, insight, confidence, essence, timing, is just a clever acronym for the components. The goal is not to replace that traditional, time and error usability evaluations, but to complement the existing evaluation techniques, and can sort of be thought of as preliminary a little bit to maybe inform those larger scale evaluations. And this is a high-level, value-driven evaluation. The elements of this framework combine to create a form used to evaluate visualization. Particularly, it's useful for evaluating multiple visualizations of the same data because it's quite a quick form to administer. And then it allows us to see the relative strengths and weaknesses, and it's meant to be used with a small group of content experts, a small group of people who understand sort of the material and the context for the visualizations. And that is where you come in. So you are my content experts. Some of you here are my co-authors on this work. You've already put a lot of time into exploring these data, exploring, analyzing harms and trials, and you're familiar with the challenges here in presenting harms from trials. Some of you are faculty or students in the Center for Clinical Trials and Evidence Synthesis. So many of you are trialists yourselves. You have experiences working with this kind of data, facing these challenges. You will appreciate and understand the purpose of this and the goal that we're trying to get at. And that's what's important for this. I think working together will make an excellent sample of experts. Maybe biased, but I think it'll be really interesting to see what we have to say. I think it will be a very informative part of our value assessment, and it will really lend itself to a strong discussion and potential future directions in further and larger evaluation. So I'm going to really sort of go through now the ICE-T assessment, sort of the questions or statements that I'm asking you to complete for me. We modified the original ICE-T to better fit the types and context of the visualizations that we're showing. So before, the statements were quite general about visualizations. They were focused on a little bit of interactivity that we don't really have. But we tried to keep the original meaning as close as possible, make it more clear for our purpose, and use the original wording in some where we thought it was appropriate. The statements are all rated with a 7-point Likert scale. So you will read the statement and then say, do you strongly disagree or do you strongly agree, somewhere in between. And then each visualization has an optional open-ended question if there's something that you struggled with or thought was challenging to understand. At the end, we'll ask everybody to rank your order of preference for presenting a summary of harms data, which one you think is best, which one you think is worst, for presenting an overall summary to people. This should take you between 45 minutes to an hour and a half to complete. It took me sort of 30 minutes, and I'm pretty familiar with the visualizations, so I hope after today, and there'll be about a 20-minute period where I can answer questions and hopefully clear up any confusion that you may have. I'm also available if you have questions to email me, and I can hopefully answer them. I know this is a big ask to do over a week. I'm really asking this to be done by Friday, April 23rd, but this is really important for supporting my dissertation work in this paper. And I hope you all find it interesting and want to give your input as well. So just as a precursor note, when completing the assessment, focus your evaluation on the contents and the approach taken by the visualization. So we really created these visualizations. We created them all using R and tried to use the same options for font size, text, color, those kinds of things, so that the purpose is then focused on the value of the contents, as opposed to things like the choices for color or the symbols used to represent. So please don't focus or think too much about those choices that were made. Those can always be changed and up to the investigators. Similarly for visualizations that present data on harms at the higher level, don't focus on the classifications that were made. So if you can see a preferred term and it's under a mid-level term that you don't think it should be, the vast majority of classifications here came from the original trialists who conducted these trials. So something that might be wrong, there's a couple that I don't necessarily agree with, but we're working with what we were given for these classifications. So that being said, here's an overview of these 4 categories, and then I'm going to go through all of these specific statements. So insight is meant to assess the visualization's ability to spur and discover insights and or insightful questions about the data, answer questions about the data. Can you see the data points in a way that's clear and makes sense? Confidence is about a visualization's ability to generate confidence, knowledge, and trust about the data. Does the visualization give you, impart a sense of trust in it and make you feel confident about what it's showing you, or is it misleading? Essence, a visualization's ability to convey an overall essence or takeaway sense from the data. Do you feel like you're getting a good sense of the harms of an intervention? Do you get a sense that you are getting all of the harms that you should be, all the information that you might want? And then timing is a visualization's ability to minimize the total time needed to answer a wide variety of questions about the data. So is the visualization clear? Is it, does a layout make sense? Are these visualizations, or is this visualization something that really requires a lot of thought, something that you have to spend a lot of time thinking about to get something out of it? Take a drink of water. So insight, again, the visualization facilitates answering questions about the data. So there's 4 statements here. This visualization exposes data for specific events. So harms at the preferred term level, dizziness, nausea. And this is, do you strongly disagree to do you strongly agree when looking at the specific visualization. This visualization exposes data for categories of events, so aggregated by system, like nervous system. I can perceive relationships between treatments and harms. Is it clear to you which harms are associated with which treatments? The visualization helps to identify unusual or unexpected but useful data characteristics or values. So is the visualization making it, drawing your attention to ones that might be interesting, harms that might be of interest, specific harms, or specific groups of harms that might be of interest? And there's an asterisk here, just for these first 2 statements to say, consider how it is presented. And this is really just because any visualization could theoretically be created to present data at any level, right? If you think back to that dot plot, which has all of the harms listed vertically, all of the preferred terms, you could create a similar plot to that by getting the number, the risk of each aggregated group and the the effect at each aggregated group, presenting the data by group. But these statements, for all of these statements, I really want you to consider them in light of how the original visualization is created and presented, not what's theoretically possible. We'll include some of that theoretical, changes that can be applied, ways that visualizations could be improved, all of that in our discussion. but just for actual comparing the value that these visualizations as originally suggested is, if you can just think of them as they are, would be great. And then timing. So this is the second domain that will be assessed. I know the acronym is ICE-T, but the way they have it set up, they do insight, timing, essence, and then confidence. So timing is about, does the visualization afford a rapid sort of comprehension for efficient browsing? So here, the visualization provides a meaningful spatial organization of the data. Do you disagree? Do you agree? Is the visualization organized in a way that makes sense and is meaningful? Timing, second statement, I can identify the key characteristics of the data at a glance. So this is, again, getting to that, is this something that you really have to work around to understand, or is this something that's clear from the way the visualization is laid out or labeled or explained? So another clarification, when we say key characteristic, we're really talking about the dimensions of harms data that are present. So the absolute counts, the effect by group, effect estimate, precision of estimate, and we're talking about the dimensions of harms that are presented in the figure. So again, it's what's presented as opposed to what could theoretically be presented, because somebody, an investigator, could choose to present a different dimension using a visualization element. You know, they could say they want color instead of representing effect size. They could say they want color to represent the count or that kind of thing. So what we really want is for you to consider this as they are presented. Essence is the domain getting at the visualization, the big picture presentation of the data. Does it provide an understanding of the data sort of overall on a whole? So the first statement, do you strongly disagree, to agree, strongly agree that it provides a comprehensive and accessible overview of the harms? Do you feel like there's everything that needs to be here, or that everything here that is here should be here, and that it's accessible, that it makes sense? The second statement then, I can understand how the different dimensions of harms data that are presented in the visualization are related to each other. So here, if you have multiple dimensions that have a relationship, does that make sense? Is it clear? So as a generic example, this may or may not be there for visualizations, but it's just sort of an example of what I mean. Increased number of events and increased precision or significance of an estimate. So if both of those domains were shown, does that make sense, how you see that and how they're represented on the figure? And then the fourth and final domain, the last statements, so statements 9 and 10, there's 10 statements overall. The visualization, elements of the visualization provide a meaningful visual representation of the underlying data for multiple dimensions of harm. So as opposed to the essence question, which looked at meaningful spatial organization, this question is getting at a meaningful representation for the specific elements, not misleading. You know, there isn't, do you strongly disagree or do you strongly agree that the elements of the scale aren't misleading you in some way? They aren't, you don't have some scale that is confusing or some color that might, some aspect of the visualization that really makes it difficult to understand or get at what the actual underlying data is representing. And then the last statement, if there were limitations like unexpected, duplicate, missing, or invalid data, the visualization would highlight those issues. So this is getting at, you know, does the visualization make it clear if you have duplicate cases or duplicate data? So maybe you have harms that have the exact same set of data. Would that be clear, or would that be something that's maybe hidden away in the visualization. So just as an example, sort of what will happen with the results, what we're going to do, and how we can use your responses to inform our assessment of value. Here's an example taken from the original paper describing the creation and testing of the iced tea. So they created these 3 different visualizations for the same data set. And once all of the responses are collected, these scores are aggregated for each of the 4 domains for each person, and then they're organized into a table like this, which really puts all 4 domains for each respondent and each visualization onto this sort of heat map like table. And this allows us to clearly see which visualizations have strengths, which visualizations have weaknesses in which domains. So we can see here the second visualization was clearly favored between the 3 that they had, particularly with regards to insight and timing. People felt strongly about that. It was good for that. And then visualization C was sort of least favored overall, lowest scores all around. People took a long time to understand, the timing, took a long time to understand the visualization, didn't feel they were getting a good sense of the overall data from it. So this can inform then our discussion and future directions when we see how all 6 of our visualizations sort of compare, where people feel strongly about something being good, doing something well, or where something needs a lot of work. So just acknowledgements then. I want to thank my advisors and committee, Alceo and Mara, author team who were involved with this and funding. And I really want to thank all of you who will complete this for me. As I said, I know it's a big ask. It's probably going to be, an hour of your time over this next week. But I'm really appreciative of it. And I think if we can get a big group of people, our people, a big group, small group of our experts to complete this, it'll really mean a lot for what we can say about the value of these. I will absolutely acknowledge you in everybody in this call in my dissertation and the paper itself as our content experts who we sourced. I won't mention by name unless you unless you ask me to do so, because I'm not collecting any identifying information from people in the Qualtrics form. So the form is set up to be completely anonymous, and it won't collect information about who's completing it. But if you want to let me know that you did complete it, I can thank you personally. Otherwise, it'll just be thank you to everybody who I asked to complete this for me. I will distribute the form via link. It is a Qualtrics form to everybody who attends today. And then if you click on the link, please do complete it and don't just leave it open through the end of the week. I'll send a reminder to everybody, regardless of if you've already started it or not, and ask for it to be completed by Friday if you are going to do it for me and help. As I mentioned, it's not human subject research, so hopefully that's okay. And yeah, so it's pretty much exactly how I expected, timing-wise, 40 minutes. So we've got 20 minutes or so for questions. Hopefully things are clear, makes sense. But if you have questions, I think we've got a couple people who can help as well. So we've got Karsten as well, who's our resident data visualization expert, turned me on to this. So if there's a question I can't answer, hopefully he can help too. And yeah, thank you for listening, and thank you for completing it in advance.
Thanks, Riaz. For those ICT components, are there some that are weighted more heavily than others, and how are you looking at those and comparing them?
So that's a good question. They are all weighted equally. So it's just basically the aggregation will be taking sort of the average of people's scores for each domain. Yeah.
Gotcha. And who are
And
the
they're all sort of positive, so there shouldn't need to be any reverse coding in the numbers. It's just whether you strongly disagree strongly disagree value of 1 to strongly agree value of 7.
Gotcha. And who are the intended audience for the results that you're planning to show?
Intended audience will really be, so we're going to be submitting this to epidemiologic reviews. This is sort of for trialists. Part of what we're doing and hoping for this paper and how we've sort of set up the results, really outlining, trying to be objective, and these are the characteristics that are shown. These are how you put them together. These are how you could modify depending on your situation. We're providing full R markdowns with examples for creation of every visualization so that any trialist who wants to create one can easily implement it in their own trial. So that's really who I guess this value assessment would be for as well. Sort of where's the value in timing and confidence it gives you about the data. But then it's also for us for when we go forwards with a more full-scale evaluation, we'll know maybe one visualization everybody hates everything about it, and so we just say based on our trialists, our evidence synthesis experts, we thought this might not be a good fit for trials going forwards as a communication tool, so we're not going to evaluate it. But that's something that will all come into the discussion.
Riaz, thank you so much for the presentation. I hope it has made it clear to most of us who are in your research in progress meeting. I just want to put this in a little bit of a context. Riaz is trying to defend by July, and this is the last bit of his data collection. So I really hope
5%.
You would really spend 45 minutes to try to help him. Again, you can imagine he created all of these. Your feedback will help to tweak all these visualizations for future projects, and that the whole field of harms research is widely open. So that's the direction he wants to go. So you're providing a lot of preliminary data as well for grants writing for Riaz in the future. He could use your feedback to either propose, modifications to existing visualization, as well as a more full-scale evaluation using more qualitative and mixed methods with different types of evidence users. As you can imagine, some of the visualization might be too challenging for a patient to understand. But then we have Karsten on the team who will be able to provide feedback in that regard. And some of this will be more interactive, as you can imagine. If you're clicking through something online, you will be able to filter out and select the type of adverse event you want to see. So that's where this fit into the whole context of, not only his thesis, but sort of, research in the future, next 2 to 3 years. So I really, really hope you will help him and provide your data to his dissertation.
Thanks, Evan. And Victoria. Yeah, so I hope, yeah, please, we still have some more minutes. So if there are no more questions, that's great. But I'm sure it's not picture perfect yet. So please do ask. And really, if you do have any questions while you are completing it, it should be set up where you can, close the survey and then come back to it. And I can try my best to be quick at responding to any questions. But we really tried to make the statements clear for our application here. And so hopefully they all make sense. And hopefully it's a little bit of fun for everybody too, to sort of critique or, really support if you feel strongly. And really do, also don't be afraid to use the broad ends of the spectrum, right? I know Likerts have a tendency to draw people to the middle, but if you really feel strongly about something, really hit that strongly agree or strongly disagree. That would be good.
Any other questions? Comments? Okay.
Okay. Well, I will send out an email with the link to everybody here today. And yeah, I'll also include my cell phone number just in case somebody wants, immediate contact and email isn't fast enough. But thank you very much.
Thank you. Great job.